Today we'll see about microbiological assay of antibiotics. So you can take the example of streptomycin or penicillin. So as you know that assay is nothing but we are going to find out the percentage purity of the given substance. So here we are performing the assay for antibiotics. So why there is a need of performing this antibiotic assay means, so whenever you got any microbial infections or bacterial infections or viral infections, you'll be taking some antibiotics into your body. So with which extent they will work on that, that means to which extent they are getting killed. So in order to know that the microbial activity or the antibiotics which you are taking, they should be very highly effective, then only we can achieve a maximum decrease in concentration of both viral or bacterial infections. So for that purpose we are going to perform the assay of antibiotics. So first we'll see about the microbiological assay of antibiotics. Example, you can take streptomycin or penicillin. So this antibiotics is based upon two methods. That means you are having two methods of antibiotic assay. One is serial dilution method and diffusion method. First we'll see about serial dilution method. So coming to the serial dilution method, the dilution of the antibiotic under test that will inhibit the growth of a susceptible organism is compared with the dilution of the standard preparation. So here we are going to do the two tests. One is the standard test, one is the test organism which you are taking. So first one is the standard preparation will be like a standard only, we are not going to add anything in that. So whatever the test microorganism we are taking, we have to keep some antibiotic solution into that, and we are going to find out whether it is working on it or not. So we are comparing the test preparation with the standard one. So how we are going to do means, we are going to do by serial dilutions of the thing. So from the knowledge of the potency of the standard, the strength of the unknown can be calculated. Dilution should be done only with the help of nutrient agar medium only. So for the dilution purposes, we have to use only the medium, that means culture medium, or you can use a nutrient agar medium. So that is serial dilution method. Next, diffusion method. So this is preferred over serial dilution method. Dilutions of the antibiotic under test and standard antibiotic preparations is made in geometric proportions. That is nothing but in the equal ratios. And we are going to prepare some plates which are prepared by pouring agar and feeding them with the test organism and allowing to set on a horizontal surface, so that the agar occupies a constant depth throughout the test. That means nothing but just you have to take a petri plate. So you have to pour the agar medium into that petri plates to constant depth. That means about 3 centimeters or 4 centimeters you have to pour the agar solution into that petri plates. Next, the plates are prepared after pouring that one. The test organism, that means whichever the microorganisms you want to grow, you have to incubate into that. So the test organism is mixed with the agar before pouring and may be applied to the surface of the medium. Antibiotic solutions are applied in cups cut in medium using a sterile cork borer about 10 cm in diameter. The cut agar disc is removed by using a vacuum device or with a pen nib. So at the center of the, after the solidification of the agar takes place in that petri plate, you have to cut it like a rectangular one, or you can make it as a small hole in the middle of that petri plate by using a cork borer, or you can cut it like a disc shape or round shape or rectangle shape, whichever you want. So cylinders of stainless steel glazed porcelain Pyrex glass or sterilizable plastic having an external diameter of 8 mm and height of 10 mm can be used, and these are slightly warm so that they sink to the constant depth when it is placed on the oven. So with the help of these things like stainless steel or porcelain, we can make holes in the middle of that petri plates, and you can remove the agar surface. Next, filter paper or cellulose disc which absorbs fixed volume of solution, and standard ceramic insulation beads also known as fish spine beads which attract a definite volume when touched on the surface of the solution. So you can use any of these one, and you can make it as a disc shaped in the center of your petri plate. Plates are left at room temperature for 2 hours to allow diffusion of the antibiotic solution throughout the medium. So in that hole which you have made in a rectangular shape or circular disc, you have to pour the antibiotic solution into that. And after pouring the antibiotic into that, you have to keep it for some time. So that it will be moving from its place to the surrounding area. So after incubation, inhibition of growth can be observed as a clear zone of inhibition. So we have already learned about this. So slowly antibiotic solution will be passing from all the sites to the other ends of your Petri plate, and you can clearly see the zone of inhibition. So from the zone of inhibition only we can clearly say that the whatever the antibiotic you are taking is working perfectly or not. So for that purpose you have to plot a graph. So a standard graph is plotted between the log concentration against the standard of zone of inhibition, and the log concentration of the test against zone of inhibition is also plotted in the same graph. And if the two lines gets parallel line, then you can say that whatever the antibiotic you are taking is showing good activity in decreasing or in decreasing the growth of your microorganisms in a specific thing. Okay, so this is the thing you have to follow. And for this, Indian Pharmacopoeia has done two assay methods. That means these are the official methods which are there in the Pharmacopoeia. So those are cylinder plate method and cup plate method, or turbidimetric method, or tube assay method. So first coming to the considerations which what we are going to take in this. So one is standard preparation. So standard is standard only. We are not going to modify anything in that. So a standard preparation is an authentic sample of the appropriate antibiotic for which the potency is determined to the appropriate international standard. So as it follows some standards, we have to maintain that standards only. So the units or μg per mg, example is streptomycin and tetracycline. You can take these values. And for the test organisms may be any of the antibiotic. You can take tetracycline, ampicillin or rifampicin. So these are some of the test organisms and assay methods, whether it is in a method or B method. That is, A method is nothing but cylinder plate method, or B method is nothing but turbidimetric method. And the medium, you can select the culture medium, either whether it is prepared with peptone or yeast extract, agar or dextrose. So coming to the apparatus, all equipment must be thoroughly cleaned before the use, and glassware should be very heated. That means you should not have any moisture content into that. And for cylinder plate method, petri dishes should be made up of glass or plastic, or rectangular glass trays are used. And for turbidimetric method, either glass or plastic tubes should be used. So here also you are having some assay designs. Two alternative designs have been recommended in Pharmacopoeia. One is 3-level or 2-level factorial assay, or 1-level assay with standard curve. So here we are also using two types of method. One is cylinder plate method, another one is tube assay method. So as you know that this method depends upon the diffusion of an antibiotic from a vertical cylinder or cavity through the solidified agar. So just now we have discussed from a solid area you are going to place some antibiotic into that, and slowly the zone of inhibition will be checked. So for that purpose you have to prepare some liquid medium. In that you have to add some microorganisms, and you have to incubate at 40 to 45 degrees centigrade, and rectangular plates are laid in the center of that petri dish about 3 to 4 mm, and solutions of concentration of standard preparation and test antibiotic are prepared in using buffer solutions, and these solutions are applied to the surface of the solid medium and the cavities in the agar, and the dishes or plates are left stand for 1 to 4 hours at room temperature at 4 degrees centigrade. This period is called pre-incubation diffusion. Then they are incubated for about 18 hours for about 37 degrees centigrade, and we can clearly see the zone of inhibition in those petri plates. Next, the second method, that is method B, turbidimetric or tube assay method. So this method is not recommended for turbid preparations, and 5 different concentrations of standard solutions are prepared by diluting the stock solution for making standard curve. A median concentration is selected, and the test sample of antibiotic solution is adjusted by dilution to obtain approximate concentration. So 1 ml of standard solution, and the solutions that are placed in each tube is in duplicate, and to each tube we have to add 9 ml of nutrient agar medium, and we have to add the test microorganisms. So you can see in the first test tube you are adding only the inoculated culture, and in the second test tube you are adding inoculated culture with 0.5 ml formaldehyde solution, and in the third one you are adding uninoculated culture medium. And these three tubes which you have prepared, they should be kept in the incubation period at 37 degrees centigrade, which is maintained for 3 to 4 hours, and after incubation you have to add 0.5 ml of dilute formaldehyde solution. Then the growth of the test organisms is measured against the standard, and they're measured in a UV spectrophotometer at 530 nanometers. So by performing this method you can clearly see the zone of inhibition. So with the zone of inhibition only you can see that whether the antibiotic is working perfectly or not. So this is about the microbiological assay of antibiotics.